Good afternoon, ladies and gentlemen. Thank you for being here this afternoon with us at our virtual platform for the scientific lectures hosted by Sharjah Archaeology Authority. Today, we are pleased to welcome Mr. Muhammad Mustafa, our guest from Egypt. Mr. Muhammad has a master's degree in heritage science, conservation science at Egypt University, July 2021. Bachelor degree in conservation from the Conservation Department, Faculty of Archaeology, Cairo University. He has many publications in scientific journals, participate in a joint project between the Grand Egyptian Museum and the professors from the Faculty of Archaeology, Cairo University in transportation and examination and by X-ray radiography, and a conservation project of 4 gilded shrines related to Tutankhamun collection from 2016 until now. Participation in a joint project between the Grand Egyptian Museum and JICA regarding documentation, examination, first aid processes, transportation, and food conservation processes of 3 gilded bed and 6 carats of Tutankhamun wood collection from 2018 until 2020. He has experience in conservation of gilded and painted wooden artifacts, conservation of more than 50 gilded and painted wooden artifacts related to Tutankhamun collection, wood science and identification of wooden species using RTI application techniques to identify the hidden inscription and the surface textures, using X-ray radiography to identify the joining technique and insect tunnel. Mr. Muhammad, mic is yours.
Thank you so much, Dr. Yazan, for your introduction. Hello, everybody. I'm Muhammad Mustafa, conservator on Wood Lab at the Grand Egyptian Museum, and I have master's degree in conservation science obtained from Egypt Japan University of Science and Technology. Really, I am so happy for being with you today, and special thanks to Sharjah Authority for their attention and their invitation today. Let's start our lecture today with entitled Closing the Gap Between Knowledge and Implementation in Conservation Science, focus on non-invasive techniques. Really, the aim of our lecture today is to help students, fresh graduates, and also young scholars to have more information further than just remedial restoration of their selected objects, which focus more on wooden artifacts as a part of my specialization, but to think furthermore or further than just remedial restoration, and to know more information about some kind of combination with a multi-scientific analytical techniques, to have a deep understanding of the manufacturing techniques and also deterioration aspects as well, how to work in just a teamwork, and how to know more information about the importance of conservation science in archaeological field. So let's start with some questions, and I will answer all these questions during our lecture today. Are you specialized in one material? Really, we are facing a big problem in specialization in many places. Maybe in our daily work, we can find many conservators who can say that we are being able to deal with all materials, and it's easy for us to do that, which is not good at all for conservators and not preferable for conservation scientists as well. It's very important to be more specialized, to read more, to focus your work more on one material, which will be very important to make a good interpretation and to know more information about how can you get valuable results behind your objects, how can you make some kind of evaluate the efficiency of using some materials and techniques, for example. So specialization is very important. Also, it's very important to work in a teamwork. There is no one can work in alone in the archaeological field, so you should have a good specialized teamwork. Consists of conservator, conservation scientist, curator, workers, technicians, and everyone who will be able to help you in your journey at the museum, for example. The second question, what are the aims of applying multi-scientific analytical techniques on the studied objects, which is very important to share more information in the upcoming slides about that. But just for your attention, you should know more information that each object could reflect different values, and it can represent tangible, natural, and intangible heritage. So each object, we can find behind this object different values like scientific value, artistic value, religious value, and many, many values. So by using conservation science and by applying some kind of combination between multi-scientific analytical techniques, it will be very important to share more information related to the object values with attention to help conservators and all sections in the museum in their daily work. And I will share more information in the upcoming slides. Also, how do conservators and conservation scientists select the appropriate way in dealing with artifacts? So it's just very simple introduction related to museums and collections, with focus more about the history of ancient Egyptian civilization, which left a very important, tangible, natural, and intangible heritage for each object from the first dynasty to the Roman period. So what is the importance of conservation science in the museums? There are many points behind, but we will summarize in 2 or 3 points. The first one is a decision making. So conservation science could help, for example, conservator to get a good decision making before applying any remedial conservation to help him to choose an ideal way in dealing with his object, to reveal more information, what kind of painting techniques, for example, binding media, gilding techniques. So it will shed light on more information which help conservators to select an appropriate way in dealing in remedial conservation with his object. Also, it could be very useful to help the conservator to know more information related to evaluate the efficiency of using new materials and techniques in remedial restoration. Also in exhibition and storage, to select an appropriate material and some kind of showcases or mounting materials, or also preventive conservation materials with a low side effects, which help our objects to stay for a long time, revealing the history behind our objects, which is very important. I think this point is very important point because, as I told you before, each object it could reveal more information and more values. And by using multi-scientific analytical techniques, for example, if you are dealing with a statue from ancient Egypt, you will have a chance to make some kind of combination between non-invasive and the multi-scientific investigations to know more information, for example, about the jointing techniques, which is very important from artistic values point of view, and also painting, gilding techniques, and many hidden inscriptions could be revealed by using multi-scientific analytical techniques. So conservation science is very important to revealing the story behind the object. Anything else? Yeah, I believe that there is many points behind conservation science in the museums. And let's have a look on this slide and asking a question. What do you think that science can help in the managing loss? As you can see here, it's one of our masterpieces. It's Tutankhamun fan. And here in the right photo, you can find this fan in a showcases at Tahrir Museum. And it's considered as one of our masterpieces. And let's say that there is no object will stay forever, but by seeking for help from conservation science, it will be very important to delay losing our masterpieces by selecting an appropriate way in handling, in what kind of materials should be in handling, also what kind of showcases, temperature, humidity, preventive conservation. So it's very important to seeking for help from conservation scientists to help us to save our masterpieces to stay for a long time, not forever. How to start? Let's start here. What do you want from your object? Are you going just to apply remedial conservation? Are you going to just applying some kind of examinations and analysis to ver some information or unexpected information? You think about that? Or you are going to get some help from conservation scientists to help you to know more information about a kind of deterioration aspects of your object, binding media of your painting layers or gilded layers as well, what kind of eternal structure of your object. So you should point your target before starting dealing with your object. Is there any samples? How many? And the ethics behind? It's very important question. You should ask yourself about this important question before starting your work. Is there any samples? If these samples are representative of all layers related to your work, how many samples? If you don't have sample, is there any ethics behind? And I advise you to know more information about ethical consideration about getting samples from JET Institute. They have a handbook, free handbook about that. And maybe you can send me a message after finishing this lecture, and I can send you more information related to this case with a PDF copy about this handbook. So it's very important about that. And also what is available techniques in your lab. In our daily work, we can meet some kind of conservators. They are asking that they will apply all materials, all investigation, all analytical techniques on their objects without good interpretation, without knowing more information about what kind of combination between these techniques to get a good interpretation and results from their objects. Also, they don't know more information about the available techniques in the labs, and what about the data that they could have from these techniques. So it's a very important question. You should ask yourselves these questions before starting your work in the object. After finishing, you should write all your information, all your thoughts in this conservation plan. And as I told you before, it's a teamwork. Your work as a museum, you should work in a teamwork. So you should discuss more with your teamwork and your team leader as well, and to figure some points to start in a good way in dealing with your object. After that, you should write brief reports about your work. What about the results? What about data you succeeded to get from your work? This is a master scene of our presentation today. Are you ready for the journey? Because as I told you in the previous slide about our colleagues in conservation, that I am daily in our daily work, I'm receiving many questions related to my colleagues, related to analytical techniques, examinations, that they don't know more information, what behind examination, why they should use, for example, a digital microscope rather than a 3D microscope, or optical microscope rather than digital microscope, or why they should use X-ray diffraction rather than a Raman microscope, for example. So it's very important. And as you can see here in this slide, and I taught an English quote talking about after great efforts, he explains that water is water. Don't waste your time about unuseful interpretation, unuseful analysis and examinations during your work in the museum and on your studied object. So it's very important to keep this slide in your mind before starting. I know that our work is focused more on some kind of combination between non-invasive, non-destructive, and destructive techniques to know more information what behind our objects. But just in brief, I will share with you 2 or 3 slides related to documentation that we can consider also some points in documentation as a primary examinations to get us primary results related to our work, photography, multispectral imaging, and I will share more information related to multispectral imaging in our case studies today, RTI technique and photogrammetry. So RTI technique here, as you can see, it's a setup of RTI technique. It's very easy technique and very useful to reveal more information about hidden inscriptions and thickness of layers and many useful information. As you can see here, the camera should be on tripod without rotating anything, without moving camera, and we can take pictures from the laptop. And the main important thing is to put black spheres here and to rotate visible light in different angles, 30 angles, 45 and 60, 90 angles, and rotate by using visible light on 330 degree around your object. So it's a very easy technique, and you have 2 software, RTI Builder just to follow his instructions. It's very easy. And after that, he can give you a software file. You can open this software file on RTI viewer with many rendering modes, so you can get more information, as I will show you in examples. It's a very easy technique. You can find more information about this technique on videos, on YouTube videos, and they have official website with more information, so it's very easy. Here, it's our basseted wooden statue from recent excavations, and you can find in the right figures. Left one is a visible figure of the face of our statue, while the second figure is related to using RTI technique, and we succeeded by using unsharp mask rendering mode to reveal hidden features around the mouse area. And also for the third figure, we succeeded to get valuable information related to the thickness of dirt as well. So it's very important, non-invasive and very effective techniques, and also it's very easy. Also, we succeeded to improve this technique by using IR light source rather than visible light source, and we succeeded to get valuable results by using this light source. As you can see, we succeeded to publish this technique in 2017, as you can see in our presentation today, and we succeeded to get valuable results by using different rendering modes. As you can see, we succeeded to get many hidden inscriptions. It's from one of our scribe books from the Old Kingdom. Also, photogrammetry, it's a very easy way to get a real 3D photo or 3D design from your objects. Just to rotate your camera and get overlapping photo and around with your camera around your objects in 360 degrees, and get, for example, overlapping photos together. And then it's easy to use many softwares. For example, we can use MeshLab, PhotoScan, and many free softwares, and it's very easy just to put your photos and follow some instructions, and after that you will have a 3D real photo of your object. It's very important techniques before starting your conservation intervention, and also before applying some kind of transportation. If you are going to make transportation for your object from place to other place, it's very important to have a record, a good record, and very easy with low cost technique for your object to know what happened before and after. So it's very important and very easy technique. After that, let's start first with some kind of examinations and analysis behind. Let's think together that we have an painted or gilded wooden object, and we are going to make some kind of examinations and analysis revealing more information about manufacturing techniques. Maybe we can start with some kind of identification of wooden species, and I couldn't be able to go more in deep in this part because it needs many informations and many workshops. But let's say that we have 3 ways in identification based on anatomic features of wooden species, some of them destructive, while other is micro or non-destructive techniques, which is very important for you to know what an appropriate way that you are going to use. If you have the ability to use non-destructive techniques, it's preferable for you. So as being conservator or conservation scientist, you should select an appropriate way before applying your examinations. For example, by using the optical microscope, it's a destructive technique, and you should cut thin sections, cross-section, tangential and radial sections to get more information about the anatomic features of each wood based on some kind of number of vessels, parenchyma cells, and rays, and many information you can get from 10 sections. And also, maybe if you have a digital microscope, you can use a handled digital microscope to get a non-destructive way in identification of wooden species to get a clear figures for 10 sections of your wooden objects without getting any samples. So it's another way as a non-destructive technique. Also, scanning electron microscope, you should have sample, but without cut in sections. So it's micro-destructive technique. So you should know more information about that. And also we have another way in identification of food and species based on chemical properties. So we have 3 ways in anatomic features and 1 way in chemical based on chemical properties, but it's a very difficult way, and I think the Getty Institute are very clever in this technique, and they have a good database of GCMS to identification of food and species, but it's still destructive technique. So if we have non-destructive way to make some kind of identification, it will be very important for you. And this is starting with examination by optical microscope as destructive technique. As I told you before, this is handled digital microscope, as I told you, and this is the way of some kind to get some kind of identification. If we succeeded to get 3 sections of wood, it will be very important for us and non-destructive techniques for us. And also it's another microscope, but it's not related to identification of food and species, but it's supposed to be that I bought this figure here with other microscopes. That 3D microscope is very important and very effective to evaluate the efficiency of your cleaning process, and also mapping, as you can see in this figure, and also high resolution scanning electron microscope, if we can use as a micro-destructive technique with samples, and also we can get a valuable quantitative results if we attach scanning electron microscope with energy dispersive, it's called EDX. So I will share with you now some examples related to identification of food and species, just for your attention to know what can we do by using different ways. The first figures, it's from destructive technique, which means that we succeeded to cut thin sections, cross section, tangential and radial sections, and this section, it's some kind of anatomical characteristics of Acacia albida. So it's kind of destructive techniques, and also we succeeded to use non-destructive techniques by using digital microscope, another kind of wood, but you can see we succeeded to get 3 sections of this wood. It's supposed to be Tamarix species, and also it's Ficus sycamorus under scanning electron microscope, but it's a degraded samples. So if you have a very degraded samples and you can deal with scanning electron microscope, it's very important. So it's very important for you as a conservator or conservation scientist to know more information, what behind your samples, what you are seeking for from your samples. It's very important. After that, it's very easy to use different ways, different techniques based on the availability in your labs to get valuable results from your objects. Let's think more with X-ray radiography, that we have this device at the Grand Egyptian Museum. It's portable X-ray radiography techniques, and these photos from JICA website, that we succeeded to get this non-invasive technique and non-destructive techniques from JICA, and it's one of our training courses on this device. It's very interesting and very important way to using non-invasive technique like X-ray radiography, which is very important to get valuable results about the jointing techniques. For example, what about the eternal structure, as well, sectus tunnels, and many informations. I will share with you some figures. It's not from our work, because our work now is still under publications, but it's clear to see here by using non-invasive technique or non-destructive techniques, more information related to mummified cat inside, with more information related to jointing techniques as well. And also here, it's from BIM Museum. It will be very important to share not also eternal structure, but also hidden inscription, any eternal, or you can reveal more information related to hidden features, as you can see here from the right figure. And also, if you think more about this figure, you can find it's a bright white on chest and also on the lady face. which gives us primary results of using heavy metals of painting materials. So maybe we can think about using lead white or like that, which has properties of fluorescence by using absorption of X-ray rays. But it's just the primary results. So it's very important for you as a conservator or conservation scientist to know more information about that. You succeeded to get a primary results. So you should think after that to make some kind of combination between some analysis and examinations to be sure about the painting techniques and gilding techniques as well. Also here, if you are interested to know more information what behind gilded wooden bits and chariots related to Tutankhamun collection, it's our poster in ICOM-CC, which was published on May 2021, with very interesting results related to the jointing techniques of gilded wooden bits, and also what about degradation in the internal structure, and also bending technique of bull and also wheels of gilded wooden chariots, which is very interesting. Also now in analysis, you should know more information. What about qualitative analytical techniques and the quantitative analytical techniques? You should know more information about that. If you are going to use X-ray diffraction, FTIR, Raman spectroscopy, it's called qualitative results. And quantitative results, for example, X-ray fluorescence, EDX, and coupled plasma. So it's very important if you have available techniques in your lab to get some kind of combination between qualitative and the quantitative techniques. For example, if you are going to make some kind of analytical study on red pigment on our any studied object, maybe we can use X-ray diffraction as the destructive techniques, or if we have Raman spectroscopy, we can use Raman spectroscopy microscope as a non-destructive techniques. So it's based on availability in your lab, but maybe we can get results about appearing, for example, hematite, but by using X-ray fluorescence or EDX, we can get concentration of each element of hematite. So it's very important to make some kind of combination between these techniques, and also it's very important to have this schedule to know more information about limitations each technique. What about behind your techniques? Is it required to have destructive techniques? It's required to have databases. It's very important to have this knowledge before starting your work. Now we are going to share some case studies from our publications. Some of the first one is related to painted wooden coffin from the late period. It's from Bani Suef storage rooms, and it was transported to the Grand Egyptian Museum in 2016. This coffin is consisting of two pieces, and you can see lid and the box, and you can see this coffin in a very bad condition with the previous conservation interventions behind. I think we don't have enough time to share all results behind this object, but you can start searching about that or sending me a text message, and I will send you the PDF of this publication. But now I'm trying to share with you some results just to know more information about the way of applying combination between some techniques to get a valuable results from your object. Firstly, we started with identification of wooden species with the destructive techniques that we couldn't be able to receive a digital microscope in this time. And the first wooden panel from the coffin box, we examined this wooden panel, and it revealed to appearing of Acacia albida. And also the second panel, we succeeded to find Ficus sycamorus. So ancient Egyptian artists used two kinds of panels in the coffin box or lid and box, which is very interesting for us. But let's think what behind about that. So that I told you before that we should work as a teamwork. This coffin is not related to important person. So the ancient Egyptian carpenters used wooden panels based on the availability of these panels in the workshop. So they used available wooden panels. They don't care about kind of wood. They used Acacia albida and connected with Ficus sycamorus by using many jointing techniques. And after that, they And after that, they covered with coarse ground layer, then painted layer. After that, we succeeded to make also identification of wooden species related to tenon and tangs, which connecting the lead with the box, and we succeeded to find its Tamarisk species. So there's 3 kinds of wood in this coffin. For the coffin body, they used Picus sycamorus, Cassia albida, but for tenon and tangus, they used Tamarisk species. Also, here, it's very important to know more information about how can you go through identification of painted layers by using non-destructive techniques to get just the primary results about kind of paintings. Here, we focus our work on using high-resolution microscope to get more information about the grains texture. So it's very important to know that behind the grains texture is a very important result, and you could get a primary result about the kind of your pigments based on the grains, is it coarse or fine, and distribution of these grains. So it's very important to study more this field to know more information about primary results related to your painted layers. Also here, now let's talk more about multispectral imaging technique. This technique that our eyes couldn't be able to see what could happen in a UV range or IR range. We are now, all of us are now trying to see invisible range, which means that visible range is from 400 until 700 nanometers. Under that, from 200 or 100 to 400, it's UV range. Above 700 until 1,200 or 1,300, it's related to IR range. So we can be able to see what happened in UV range and IR range. So multispectral imaging technique is very effective and non-invasive techniques to know more information what happened in UV range or IR range. And there are many references behind that there are many painted layers have some kind of properties, some kind of fluorescence properties. For example, on Horace tail, if you can see here, you can find it's a bright white invisible induced luminance. So it's a good indication related to Egyptian blue. So Egyptian blue has a fluorescence properties, invisible induced luminance or IR fluorescence. So if you are going to get clear figures from IR in IR fluorescence, it's very easy to use a full spectrum camera and LED visible light. Also to put some features, some filters on your camera to cut visible range, UV range, and to give us more information about clear photos, what happened in IR fluorescence. Also, the third figure is related to IR false color, and it's this technique was applied by using Photoshop, and there are many videos on YouTube talking about this case. So it's a very easy technique. The case now is that you need to know more information what happened in UV range or what happened in IR range. If you are going to know more information about UV reflected or UV fluorescence results on your objects, also in IR range is the same way. So the case is just to using full spectrum camera and switching between many lights like IR light or UV light or visible lights based on the availability and what do you know, what do you want to know from your painting techniques, and also using many filters about related to just cut UV range or pass your UV range and cut visible range, or pass IR range and cut visible and UV range. So it's just your work and it's very easy way. Also after that, we succeeded to find that it's from primary results, maybe it's related to Egyptian blue, and we succeeded to get more information by using X-ray fluorescence with all elements related to chemical structure of Egyptian blue. But let's say now if you are going to use an X-ray fluorescence, it's very important not to use one spot, because it's a spot analysis. So it's not a good way to just get a spot analysis in one shot and to say that I have XRF chart. It's not good at all. You should have at least 3 or 4 spots in the same area and make some kind of average and it's very important to have average from these spots to get a good results from X-ray fluorescence, and also EDX, and also any spot analysis, because maybe we can find some kind of changes from area to another in the same objects. Some kind of objects, maybe if this spot area contains more deterioration aspects or contamination from other pigments, so it's called reflected. It's going to be able to reflect a reality results from your work. It's very important to have 3 or 4 spots from the same area. After that, we succeeded to find another kind of blue on the body of goddess Nephis, and by using a microscope, it's very important that we find it's not coarse grains, so it's fine grains with some kind of contamination, if you can see from yellow pigments, and also unknown dirt layer. And also by using false color techniques, there is no reddish evidence, so it's not Egyptian blue. So now we don't have Egyptian blue. And also by using X-ray fluorescence, we don't have any elements related to using cobalt blue. So we have a strong suggestion about primary results related to using azurite. And the second spot, that if you can see here in left figure about a greenish line on the decorated body, that ancient Egyptian didn't intend to use this technique, but by using X-ray fluorescence, maybe it's related to using malachite. But why ancient Egyptian artists intended to use malachite just on one line in the decorative body? Really, we have a strong suggestion about this case, that it was this decoration was applied on the outer base of the coffin, so it's so close to the humidity in the tomb. So by using azurite with humidity, you can find some kind of degradation. So we can get degradation of azurite and transferred to malachite. So we succeeded to find malachite here as a kind of degradation of azurite. So if you are not specialized enough, if you don't have more information about kind of pigments and kind of the degradation of your pigments and also the history of your pigments, you couldn't be able to get a good interpretation. You should be specialized enough in one materials to get a valuable result at the end. The second case study is from our recent publications, Archaeometric Study and Conservation of a Goddess Basseted Statue. It's from also recent excavation, Saqqara. And here we started with some kind of identification of insect species. The left figure is related to carpet beetle, spider beetle, and also we succeeded to find lizard skeleton inside the pedestal of this statue. And also we succeeded to use microscope to make identification of textile species, because let's think that this manufacturing technique of this statue consists of wooden base covered with textile, and then coarse ground layer, fine ground layer, and then painted and gilded layers. So it's very important to make some kind of interpretation of each layer. So identification of textile species revealed more information about using linen, and also it's very easy for us to know more information related to deterioration aspects as a case of insects attack. And also if you are going to make some kind of cross sections, it's considered as destructive, but it could be semi-destructive techniques if you are clever enough and you don't need to have a high resolution microscope. As I told you before, if you are clever and you know more information what about the data you are going to get from your objects, it's very easy even to use USB microscope to get more information. These photos are from USB microscope, which is very important and very interesting to use the available techniques in your lab, because this object, this analytical study were applied in a storage room with limitation in available techniques. So if you are specialized in wooden artifacts and you are going to put your hand on a good technique, it's very important for you. Even if you don't have available analytical or examination equipments, maybe you can use even USB microscope to get more information and valuable information related to the sequence of layer, for example, and also the thickness of each layer. Here you can get more information, as I told you in the previous slide, related to ancient Egyptian coffin in identification of wooden species and all, and you can find the fluorescence here, invisible induced luminescence, which gives more information related to appearing of Egyptian blue. The second photo here is UV figure, and the last photo is related to IR figure. So UV figure and IR figure is very important for us to get valuable information. Related to UV figure, it's very important to get more information related to some kind of varnish. So you can find some areas here are a bright white related to using varnish. And we applied this technique by using full spectrum camera, UV light source, and also some filters. These filters helps us to cut UV range and IR range. So we are going just to know more information about the reflection of UV. We need the camera to see what we are going to see in our eyes. So it's very important. Also IR figure with the same technique, using IR source, a full spectrum camera, filter to cut the range of UV and also visible, and also, sorry, IR range. So it's very important. This technique is very important to reveal more information about hidden inscriptions. Let's start with the last study. It's a gilded wooden statue from the Ottoman period, and it's an Islamic object. And really, this object, we have a story behind this object, because this object was attempted to smuggle out of Egypt, and our police officers succeeded to get this object before traveling abroad. And I was lucky to start looking for this object and starting some kind of examinations and analysis, which was very interesting results and decoration techniques as well. This statue consists of two pieces, body with black face and decorated with multi-colors on clothes and base, which was designed to rotate the body of a statue 360 degrees. It's very interesting to share with you that the decoration technique of the statue. The statue is made from wooden base, covered with coarse ground layer, then fine ground layer, or whitewash, and after that applying gilded layer. And very interesting technique is applying a painted layer on the gilded layer. So it's very unique object that we couldn't be able to find it before. We succeeded to make 3D modeling by using photogrammetry. As you can see here, we succeeded to take overlapping photo, as I shared with you in the previous slides, with Mesh Lab software. So we succeeded to get a primary result for 3D generated photo of photogrammetry, and also by using use microscope. It's a very simple and effective way as a non-destructive technique to get more information about the manufacturing technique. As I told you before, the painted layer was applied on the gilded layer, which is very unique. And also we succeeded to find more information about the sequence of layer, wooden base, on the 5 figure, if you can see, wooden base covered with coarse ground layer, and maybe after that fine ground layer, and then gilded layer, and after that applying painted layer on the gilded layer. I couldn't be able also to share all results behind, but starting with identification of wooden species, it's our hand cutting. So you should be clever to get these reference samples for you. We succeeded to get by using thin sections to get these sections, cross section, tangential, and radial section. And it's one of our softwoods and it was identified as a pine wood species. Also, identification of previous conservation intervention by using UV or multispectral imaging techniques. Here, if you are going to take a good figures for gilded object, you couldn't be able to use just camera. If you can see the left figure, it's just by using camera, but the middle figure by using camera with a polarizing filter. So you should use polarizing filter if you are going to get a good result from gilded objects to prevent diffusion of gilded layers. And by using visible light with polarizing filter, it was clear to find previous conservation interventions areas. And also by using UV images, it was clear to find that it was quenching fluorescence rather than the surrounding area, which means that we have here previous conservation interventions. After that, we succeeded to use EDX as a quantitative analytical technique to get elemental results about the previous conservation interventions, which means that we succeeded to use, they used cadmium. If you can see here, cadmium is a main elements, which revealing to using cadmium
Mr.
yellow.
Muhammad,
Ayo.
sorry for bothering. Can you please try to finish faster?
Yeah, just 5 minutes, if I can.
Okay.
Yeah, it's okay. Thank you. So they used cadmium yellow as a previous conservation interventions. Also, by using multispectral imaging techniques here, it's the face of our statue, and for first look, we couldn't be able to find any previous conservation interventions. By using UV figure, we surprised to find many previous conservation interventions. As a whole face of this statue, if you can see, it's more a bright white. It's related to using some kind of varnish, but quenching fluorescence of around the mouse related to using previous conservation interventions. So by using EDX, we succeeded to find that the original black layer, black painted layer, was made of carbon-based pigment, while the previous conservation interventions related to using titanium-based pigments. Also succeeded to make identification of varnish by using FTIR ATR as a non-destructive technique. So maybe we can use FTIR in two ways: destructive techniques by using in a KBR mode, or ATR. So it's based on availability and what do you want from your objects. It's very important. So by using FTIR ATR, we succeeded to find that they used an animal glue as the binding media of a black painted layer, and also varnish called shellac. We will finish with just 2 slides or 3 slides just to show you more information about bad conservation interventions without for help from conservation scientists. It's a well-known painting from Face of Jesus. It's from church in Spain with untrained, not trained conservator. So the result was very disappointing. But from marketing technique, from marketing way, they succeeded to use this way to make a good marketing of these shares. And you can see that they succeeded to put this botched restoration intervention on wine bottles, and on mug, and on everywhere. So they succeeded to attract many tourists from several places to this church. So from marketing point of view, it's a very effective way, but from conservation or conservation science point of view, it's very bad way. I succeeded to finish, and if you have any questions, I'm ready, and thank you so much for your attention.
Yeah, thank you so much, Mr. Muhammad, for this good lecture and important information you gave us during your lectures. Dear audience, please, can you write your email and name in the link we sent it to the messages, and also fill the questionnaire. Also, we sent it to the messages. We have questions from Mr. Muhammad Hawash. Mr. Muhammad, mic is yours.
Yes, and feel free to ask your question in Arabic or English. It's okay.
السلام عليكم.
وعليكم السلام.
السلام عليكم.
اولا نشكر اداره الشارقه، ونشكر الدكتور محمد مصطفى. بسم الله ما شاء الله، بسم الله ما شاء الله عليه. يعني المحاضره محاضره ممتعه وتحمل الكثير من المعلومات، لكن كل برانش او كل جزئيه او كل فرع هو تناوله كان يحتاج كثير من الاسهاب، لان محاضره واحده في موضوع الايدنتيفيكيشن للاخشاب والعلاج والصيانه ومعرفه التدخلات السابقه لا يكفي محاضره واحده. لكن هو طبعا يعني تعودنا منه دائما الاجتهاد، فوفى بقدر المتاح من الوقت. انا عشان ما اطولش على حضراتكم، انا عندي بس 3 استفسارات للدكتور محمد. دكتور محمد، بعد اذن سيادتك، انت جيت اتكلمت في الشرايح الاولى عن النون ديستركتيف ميثودز، وكنت ذكرت ان اليو اس بي ميكروسكوب اثناء الايدنتيفيكيشن للخشب ممكن يكون من النون ديستركتيف ميثودز. هو السؤال هنا، هل اليو اس بي ميكروسكوب انت بتعمل سامبلنج او بتعمل اعداد لشريحه وبعد كده تفحصها بيه، ولا بتستخدمه مباشره على الخشب؟ لان لو بيستخدم مباشره على الخشب يبقى كده خلاص بقت فعلا نون ديستركتيف ميثود. اما لو في بريبريشن لسلايد او في اعداد لشريحه، خاصه على حد علمي ورغم ان الاخشاب مش تخصصي، ان الاخشاب اثناء الايدنتيفيكيشن بتاعها بتبقى انت عايز تجيب قطاعات معينه بترصد فيها شكل الخشب فيزيكالي على الاقل، علشان تقدر وتقارنها بعد كده عشان تقدر تعرفها. فعلى الاقل انت بتحتاج تعد سلايد او تعد شريحه في قطاع معين في الخشب علشان تقدر تعمل ايدنتيفيكيشن. لو بالطريقه دي بقت ديستراكتيف. طب لو انت قلت ان هو نون ديستراكتيف، يبقى معنى كده ان احنا هل متاح ان احنا نستخدمه مباشره دون اعداد شريحه؟ ده السؤال الاول. ممكن سيادتك تجاوبه واكمل السؤالين التانيين بس عشان من
تمام يا دكتور. طبعا انا بشكر حضرتك على الحضور طبعا، وبشكر حضرتك على الكلام دوت، وشرف ليا طبعا حضور حضرتك النهارده.
الله يسعدك.
وطبعا انا عارف طبعا ان احنا اتكلمنا في مواضيع كتير قوي النهارده، ولكن كنت بحاول ان انا اخد الحضور النهارده في رحله كده بسيطه، نقدر نتعرف بس على الوضع بتاعنا ممكن يبقى ماشي ازاي، وبعد كده ان شاء الله يبقى في محاضرات اكتر تخصصيه، يعني اعتمادا بس على وقت المحاضره، فحاولت اجتهد فيها على قد ما اقدر يعني. دي في البدايه. الحاجه الثانيه، حضرتك ذكرت القصه بتاعه تعريف الخشب. انا ذكرت حاجتين في استخدام الميكروسكوب في تعريف الخشب. هنستخدم الاوبتيكال ميكروسكوب، الميكروسكوب الضوئي العادي ترانسميتد لايت، ده بيبقى ديستراكتيف تكنيك، ده بيبقى تقنيه متلفه. ليه؟ لان احنا بنضطر ان احنا نقطع شرايح، بنقطع سين سيكشنز، بنقطع السين سيكشنز دول اللي هم بيبقوا ايه؟ بيبقى القطاع الكروس سيكشن والتانجنشال سيكشن والراديال سيكشن، القطاع الطولي والعرضي والمماسي. تمام؟ وبنبدا ناخد الشرايح اللي احنا قطعناها دي تحت الميكروسكوب، بنبدا نعرف الخشب بناء على الخصائص التشريحيه بتاعت الخشب. وده شغل بوتاني اكتر شويه، ولكن احنا طبعا زي ما حضرتك عارف ان احنا شغلنا شغل بيني شويه، فبنحاول على قد ما نقدر ان احنا نغطي بشكل كبير بعض التخصصات عشان تقدر تفيدنا في شغلنا. فلو استخدمنا الاوبتيكال مايكروسكوب بالشكل ده، ان احنا قطعنا وفحصنا تحت الميكروسكوب، ده بيبقى ديستراكتيف تكنيك، ده تقنيه متلفه. خلاص احنا خدنا عينات وقطعنا وفحصنا تحت الميكروسكوب، فدي كده تقنيه متلفه. طيب لو استخدمنا بقى النقطه الثانيه اللي انا اتكلمت فيها، اللي هو البورتابل هاي ريزوليوشن ديجيتال مايكروسكوب. في بعض الميكروسكوبات دلوقتي ديجيتال وهاي ريزوليوشن زي اللي انا عرضتها في البرزنتيشن. دي حضرتك الميكروسكوب نفسه بيبقى بورتابل، وحضرتك بتروح بيه للقطعه نفسها من غير ما تاخد اي عينات، وتحاول تطلع بالهاي ريزوليوشن مايكروسكوب دوت الثلاث قطاعات بتوع الخشب. هي الفكره كلها ان حضرتك تبقى وانت داخل تعرف خشب، لازم تبقى عارف انت بتدور على ايه. يعني تبقى عارف ايه هو الكروس سكشن، التانجنشال، الراديال، ايه اللي انت عايزه من كل قطاع. فلو قدرت تعمل ده بالهاي ريزوليوشن بورتابل ديجيتال مايكروسكوب، ده بيبقى نون ديستراكتيف تكنيك، لان احنا ما بنقطعش عينات، احنا بناخد الميكروسكوب وبنروح بالبورتابل هيد لغايه القطعه نفسها، وبنحاول نطلع الثلاث قطاعات من القطعه. انا ما جبتش سيره بقى طبعا اليو اس بي مايكروسكوب في القصه بتاعت التعريف، ولكن احنا كمان قدرنا ان احنا نستخدم اليو اس بي مايكروسكوب كتقنيه غير متلفه في عمليه التعريف. بس انا طبعا ما قدرتش ان انا اشير لبعض النتائج دلوقتي، لان الموضوع تحت الدراسه. هي الفكره كلها بس ان الموضوع محتاج صبر كتير، ومحتاج ان حضرتك تبقى زي ما انا قلت، محتاج تبقى عارف ايه اللي انت محتاجه او اللي انت عايزه من الثلاث قطاعات بتوعك. وبالتالي بتبدا حتى باليو اس بي مايكروسكوب، لو حضرتك شغال في مخزن او شغال في مكان ما فيهوش تكنيكس افيلابل، تكنيكس موجوده، ممكن حضرتك بتروح باليو اس بي مايكروسكوب نفسه للقطعه، وتحاول على قد ما تقدر ان انت تطلع القطاعات بتاعته. ده برضه بيبقى نون ديستراكتيف تكنيك. هي الفكره كلها احنا خدنا عينه وقطعنا وفحصنا فيبقى تقنيه متلفه، ولا احنا رحنا بالميكروسكوب نفسه لغايه القطعه من غير ما نقطع ومن غير ما نشوف السكشنز، فبالتالي بقت التقنيه غير متلفه. بس هي دي الفكره، يعني هي دي الحكايه.
تسلم يا دكتور محمد، بارك الله في حضرتك. بس كده معنى كلامك ان ده بيعتمد على شكل العينه، يعني انا لو هشتغل بالبورتابل ميكروسكوب او اليو اس بي ميكروسكوب لازم يكون على الاقل الثلاث قطاعات ظاهرين عندي. معنى كده ان انا ممكن لو العينه تالفه او فيها كسر هيبقى اسهل ان انا اقدر اكشف الثلاث قطاعات بتوعها، صح؟
مش شرط تبقى تالفه او فيها كسر. طبعا هقول لحضرتك على حاجه، قصه النون ديستراكتيف تكنيك دوت، يعني الطرق الغير متلفه باستخدام الميكروسكوبات كمان، بتبقى محتاجه صبر كتير، وبتبقى محتاجه حظ برضو، يعني يعني ان حظك يبقى كويس مع القطعه.
بالظبط
تمام،
كده.
حظك يبقى كويس مع القطعه ليه؟ لان مش دايما حضرتك بتلاقي القطعه خشب بس، وخشب ظاهر فيه كل القطاعات. في
بتبقى
الغالب
اثار مركبه.
في الغالب زي ما حضرتك عارف طبعا، زي ما حضرتك قلت دلوقتي، هي اثار مركبه. فبعض الاحيان حضرتك بتلاقي بعض الاجزاء من القطعه مش موجود عليها طبقه التحضير او طبقه الالوان، وحالتها تسمح بالنون ديستراكتيف تكنيك. يبقى ساعتها احنا ممكن نعمل البورتابل تكنيكس، فبالتالي هيبقى نون ديستراكتيف. لكن ممكن تبقى العينه ظاهره قدامنا بس الديجراديشن فيها عالي جدا، فلو حضرتك استخدمت حتى الهاي ريزوليوشن مايكروسكوب، مش هتقدر تطلع الخصائص التشريحيه بتاعت الخشب، لان العينه متحلله. فبالتالي هنستخدم تكنيك تاني خالص، او نستخدم حتى الجي سي ام اس، ونعتمد على الخواص الكيميائيه للخشب، لان كل نوع خشب ليه خصائص كيميائيه، وفي قصه دي قصه كبيره ثانيه. فهي الفكره كلها في كده، هل الكلام ده متاح من خلال القطعه نفسها تسمح لحضرتك بكده، والتكنيكس اللي موجوده عند حضرتك في المعمل تسمح بكده ولا لا؟ طيب الكلام ده مش متاح عندي، بس انا عندي عينه اوريدي واقعه من القطعه. طب العينه اللي واقعه من القطعه دي، هل ابعاد العينه دي تسمح ان انا اشوفها تحت الديجيتال مايكروسكوب؟ لو ما تسمحش، طب هل تسمح ان انا احطها تحت السكاننج الكترون مايكروسكوب، ودي تبقى برضه نون ديستراكتيف واي، لان انا ما قطعتش، هي العينه كده كده اوريدي واقعه، وانا خدتها حطيتها في السكاننج الكترون مايكروسكوب وحاولت اجيب الثلاث قطاعات بتوعها. هل لو انا قدرت اعمل كده هيبقى الوضع ايه؟ هيبقى نون ديستراكتيف. هي الفكره كلها بتعتمد على ايه؟ حاله القطعه اللي قدام حضرتك، والافيلابل تكنيكس اللي موجوده عند حضرتك في المعمل. ده اللي انا كنت بتكلم فيه، ايه المتاح وايه اللي حضرتك تقدر تعمله بالمتاح عندك.
تمام يا دكتور، بارك الله في حضرتك. السؤال الثاني بقى يا دكتور محمد، انت حضرتك برضو صنفت الطرق بتاع الاناليسيز الكوانتيتيف والكواليتيف، وده تصنيف بسم الله ما شاء الله عليك، يعني اصبت فيه اصابه كبيره. لكن في نقطه، لما انت اتكلمت عن الاي ار والاكس ار دي، ذكرت ان هم كواليتيف، واتكلمت عن الايدكس او الاي سي بي، الكلام ده كله اللي هي التحاليل العنصريه، انها كوانتيتيف، دي لا خلاف عليها انها كوانتيتيف، سواء الاي سي بي او اللي هو تفاضل الاشعه السينيه، برضو كل دول ما فيش خلاف عليهم. هي الفكره بس في الاكس ار دي او الاي ار، لما حضرتك ذكرت ان هم تحاليل كيفيه فقط، هنا بس هتبقى في اشكاليه. يعني دلوقتي مثلا على سبيل المثال الاكس ار دي بيعرف لي الماده كمينرال كمعدن، فلو انا جبتها مثلا اي سي بي هيديها لي اليمنت، او اكس ار اف هيديها لي اليمنت برضه. ففي بعض الاحيان بتبقى المعادن مشترك بينها اليمنتس كتير، يعني انت مثلا لو بتعرف جبس وتعرف جير، هيطلع لك مثلا اكسيد الكالسيوم، سواء انت كده مش هتعرف هل هو جبس ولا هو جير. فبنبقى محتاجين ان الطرق الكميه او الطرق الكيفيه اللي هي الكواليتيف زي الاي ار او الاكس ار دي، ان احنا نقدر نحصل منها على نسب على الاقل يمكن اعتبارها على انها نصف كميه. يعني مثلا على سبيل المثال لو انت معاك وزن العينه والكلام ده، فممكن نقدر نحسب او نحصي مثلا المجموعات الوظيفيه من خلال الاي ار، او على حسب طريقه اعداد العينه لو هي مثلا مطبوخه مع بروميد بوتاسيوم او على عينه سائله. وكذلك الاكس ار دي برضه في اثناء دخول العينه في الديفركتوميتر ممكن يتم وزنها، ويتم برضه معرفه بشكل مش هنقول كمي بنسبه كبيره ودقيق بنسبه 100%، ولكن بشكل نصف كمي. لان لو اعتمدنا بس على التحاليل العنصريه ممكن اعتقد ان هي هتدينا نتائج اي نعم دقيقه، ولكن مش هتقدر تنسب هل زي ما قلت لسيادتك كده، هل اكسيد الكالسيوم ده مثلا او الكالسيوم اللي طالع ده بتاع جير ولا بتاع جبس. هتلاقي العناصر المشتركه ما بين معادن كتير، هتلاقيها كتير. فيعني اتمنى انك تفيدني في النقطه دي، هل يمكن بالفعل اعتماد ان الطرق الكيفيه زي الاي ار والاكس ار دي يمكن اعتبارها على انها طرق نصف كميه، ويمكن تحقيق ده بالفعل في الواقع ولا لا؟ انا اسف على الاطاله.
لا ما فيش مشكله طبعا، تحت امرك يا دكتور. بص اقول لحضرتك على حاجه، هي الفكره كلها اللي انا كنت بعرضها في فكره التحاليل الكميه والكيفيه. انا طبعا لما جيت ذكرت في البرزنتيشن، انا اللي اتكلمت فيه اللي هو احنا ما ينفعش نعتمد على تكنيك واحد يا جماعه، ما ينفعش نعتمد على جهاز واحد بس في التحليل. تمام؟ لو احنا قادرين اللي احنا نقول ان احنا والله يا جماعه عندنا في اللاب بتاعنا اجهزه بتطلع لنا كواليتيتيف واجهزه بتطلع لنا كوانتيتيف، وقادرين نعمل الربط ما بينهم، ده هتبقى حاجه ممتازه جدا. ليه؟ انا دلوقتي مثلا زي ما شرحت في البرزنتيشن، انا استخدمت الاكس ار دي، الاكس ار دي طلع لي الهيماتايت، الاكس ار اف طلع لي نسبه كل عنصر في الهيماتايت عامله ازاي، فانا عملت ربط ما بين الكمي والكيفي. قصه ان انا اقدر استخدم الكمي ان هو يديني حاجات نص كيفيه، او القصه دي بتبقى ماشيه ازاي؟ هي الفكره كلها احنا عندنا امكانيات ولا ما عندناش؟ هنعرف نعمل ربط ولا مش عايزين؟ الفكره كلها هل حضرتك كباحث او انا كباحث، هل احنا مهتمين في الدراسه بتاعتنا ان احنا نعمل نون ديستراكتيف تكنيكس اصلا ولا ديستراكتيف تكنيك؟ هي دي الفكره. يعني مثلا انا لما استخدمت في التابوت الخشبي في الشغل بتاعنا، لما استخدمنا عليه، انا كان الهدف بتاعي والتارجت بتاعي من البدايه ان انا اعمل ايه؟ ان انا اعمل نون ديستراكتيف تكنيكس. ما انا كان عندي اكس ار دي وكان عندي اف تي اي ار، بس حبيت ان انا اربط بتكنيكس غير متلفه عشان اطلع نتائج بقى ايه، انا ما بقولش عليها نتائج نهائيه، او ما بقولش عليها والله ده طلع لي هيماتايت، لا، انا حتى وانا بكتب في الابحاث بتاعتي بقول ايه، ربما يحتوي على هيماتايت. طب انا بقول ليه كده؟ لان انا ما دعمتش بكواليتيتيف اناليتيكال تكنيك. طب ما دعمتش ليه؟ لان انا كنت عايز اخش في سكه ثانيه اللي هو انا بدعم بالنون ديستراكتيف تكنيكس. هي دي الفكره. لكن ان انا استخدم كوانتيتيف بس، ان انا استخدم اكس ار اف بس او ايدكس بس واقول والله لا ده ممكن يطلع لي بقى نتائج، لا طبعا الكلام ده كله مش صح. والكلام ده بيلخبطني كتير جدا، لان حتى عندنا في الالوان كمان في لخبطه كبيره. في بعض الاحيان ممكن يبقى عند حضرتك على سبيل المثال ازرق، اللون الازرق لو جيت فحصته تحت الاكس ار اف على سبيل المثال ونسبه السيليكا قليله، فحضرتك ممكن تشك ان ده ممكن يبقى ازرق مصري، ممكن تقول والله ده ممكن يبقى ازرايت. فالموضوع هيختلط على حضرتك، لان الكيميكال ستراكشر ممكن يبقى قريب شويه في الاليمنتال، يعني العناصر قريبه شويه من بعض. فبالتالي انا ما اقدرش اعتمد على نتائج زي نتائج الاكس ار اف ان انا اقول والله الاكس ار اف هيديني النتيجه دي فخلاص انا يا جماعه ده اكس ار اف، ده سوري ايجيبشن بلو. لا، ممكن ما يبقاش ايجيبشن بلو. هي دي الفكره. الملكايت والازورايت ممكن لو في ديجراديشن زي ما انا قلت حاصل، الملكات والازورايت قريبين جدا من بعض، فما ينفعش ان انا اعتمد على سبوت اناليسيس من الاكس ار اف واقول والله لا انا كده ظهر لي يا جماعه ازورايت، لا انا كده ظهر لي ملكايت. ده هيعمل ديفيوجن جامد جدا وهيعمل مشكله كبيره جدا. ولكن انا لو استخدمت حاجه زي الرامان مايكروسكوب على سبيل المثال، وهو برضه سبوت اناليسيس، ولو متاح عندنا دي تبقى حاجه كويسه، وفي امكانيه ان انا اعمل سكان اريا فاقدر اطلع الافريج واقدر اطلع نتائج ممتازه، فهيديني كواليتيف ريزلتس. هيقول لي لا والله حضرتك عندك دلوقتي ده ازورايت، ده ملكايت، ده ايجيبشن بلو. بعد كده انا بقى على حسب المتاح عندي ممكن استخدم ايدكس، ممكن استخدم اكس ار اف، ممكن استخدم اي اليمنتال اناليسيس، واقول والله لا انا عندي بقى دلوقتي نسبه النحاس طلعت قد كذا، نسبه السيليكا طلعت قد كذا. ممكن كمان اعمل بقى حاجه يعني ادفانسد لو انا حابب ان انا اتعمق شويه في القصه دي واقارنها بعينه قياسيه، فاقول والله لا ده انا لما جيت قارنت بكنترولد سامبل من الكواليتيتيف والكوانتيتيف، ده انا قدرت كمان اعرف نسبه الديجراديشن اللي حصلت عندي في اللود. فهي اللعبه كلها فين؟ حضرتك محتاج توصل لايه؟ ايه المتاح عندك؟ وما ينفعش تعتمد على تكنيك واحد بس.
احسنت يا دكتور محمد، بارك الله في حضرتك. انا كان في سؤال ثالث بس انت جاوبته ضمنا في الاجابه اللي حضرتك اوردتها دلوقتي. أنا سعيد بالمحاضرة، والمحاضرة بالفعل من باحث قوي وأضافت للواحد كتير، وبإذن الله يبقى فيه محاضرات تانية تفصيلية، لأن الموضوع كل نقطة بالفعل محتاجة محاضرة يعني. وبالتوفيق إن شاء الله يا دكتور محمد، ونشوفك دايما يعني في محاضرات وأبحاث مثمرة دايما بتميط اللسان عن الأسئلة العلمية اللي بتبقى شائكة. وده عهدنا دايما بيك، وربنا يوفق دايما إدارة الشركة في اختيار الباحثين أصحاب الملاءة أمثال حضرتك كده، وبالتوفيق والسداد يا رب دايما. شكرا.
ربنا يبارك في حضرتك يا دكتور، وشكرا لحضرتك على الأسئلة القيمة. شكرا جزيلا.
دكتور، في عندنا سؤال على المسجات. Does the work of preserving antiquities require that the workers on them to be from certain specialists or various specialization, including specialists in restoration, Egyptology, and others?
Let's talk more. You should have a specialized person in your museum work. Even if you are going to make transportation for the selected object from the place to another, you should select specialized workers to do this case. We are responsible for preserving our artifacts to stay for a long time, to be a witness of our civilization and to reflecting several values behind. So it's very important to have a specialized person behind. In any case, in preserving artifacts in our museums or in archaeological field, we should have teamwork. This teamwork is consisted of conservators. Conservators are playing an important role in this case. If you are going to make a transportation for the object from place to another place, it's very important to have a good conservator who can make some kind of first aid materials to make stabilization for the friable layers before a transportation, and to get advice about an appropriate way of dealing with the object before transportation. For example, I just put an example to get your attention about different specializations. Also, curator is very important to show with you more information what behind the object, what about Egyptologists. From Egyptologist point of view, if there is any very important inscriptions need translation or so on, so curators playing an important role in the museum work. Also, conservation scientists is very important to use non-destructive and non-invasive techniques to save your object before and during transportation, and also after transportation to apply indeed multi-analytical investigations to get valuable results related to your object, which reflecting more information related to artistic values, and help conservators to select an appropriate way in dealing with these objects. Also, interpretation of previous conservation interventions or deterioration aspects, or also helping conservators to evaluate the efficiency of using new materials and techniques. So we should work as a teamwork, but if you are working in museum or archaeological work, you should think about specialized person, well-qualified person in their work. It's very important. You should select a good person for your journey as a museum.
Yeah, thank you, Dr. Muhammad, for these answers. Thank you for, there is no any more questions. So thank you so much for your good lecture. Really, it's nice lecture. I like it, and good information, important information, and you gave us. Dear audience, thank you so much for your listening. Inshallah, see you next time in the next lecture. Thank you so much.
Thank you.